Your sample. Welcome back, everyone, and thanks for joining us for this next session about the newly developed clinical care guidelines for Sanfilippo syndrome. This project grew out of a collaboration between Cure Sanfilippo Foundation and the Sanfilippo Children's Foundation in Australia. I'm Dr. Cara O'Neill, Chief Science Officer and co-founder of Cure Sanfilippo Foundation, and mom to my daughter Eliza with Sanfilippo Type A. And with me today is someone who probably needs no introduction to this group, Dr. Joseph Muenzer, Professor of Pediatrics and Genetics at the University of North Carolina School of Medicine. Thanks for joining, Dr. Muenzer.
Well, thank you for the opportunity to talk in the guidelines. They're a really important development for helping improve the outcome of patients with Sanfilippo syndrome. Thank you for letting me be here.
Yeah, thanks for being part of the steering committee as well. And also want to recognize our other steering committee members who were instrumental in the development of this guidance. I thought one way we could start today is just with an anecdote that so many families express to us that they hear at the time of diagnosis, and that's the message that they're told to take their child home and love them, that there's really nothing that can be done. And I know we're all out to change that message ultimately, but you've been involved with clinical guideline development for many diseases, and I wondered if you could speak to the importance of guidelines and how they make a difference.
As we all appreciate, the Sanfilippos are really rare, very rare in terms of frequency, and most individuals rarely, most geneticists, most physicians rarely see them. And so this concept that, we have nothing to do is really incorrect. There's a lot of things we can do to improve the quality of life. Especially as new therapies are starting to be developed, we have a chance to really enhance how the kids do. And so guidelines are important because we all are sort of biased by our own really experience. Sometimes we have one bad experience and everything is clouded. So guidelines are really developed by a variety of individuals, physicians, geneticists, other specialists who have experience with this disorder and can really come to a composite or just a unique collection. And so it's so important to really highlight what needs to be done, what disease process is really going on. And that's, I think, the key for me is to show people, here's something that can happen in Sanfilippo. It doesn't always occur. Again, as we all appreciate, Sanfilippo was primarily a neurologic disease with cognitive issues, but there can be significant physical involvement that we need to sort of be aware of that we can impact sometimes more than we can the cognitive currently. So it's really this awareness, and for families to sort of sense what they should do maybe if they're having a particular issue. So I think guidelines are important. They're guidelines. They're not written in stone. They should be not taken absolutely. They need to be in context with what happens going on in the child's life at the time. You try to use these guidelines.
Yeah, thank you. When we were talking about this as a group, another thing that really came up was increasing consistency of care and access to the knowledge of what would be considered, kind of consensus of what good care looks like in Sanfilippo. Again, understanding everything needs to be taken individually, but for those in remote places or who don't have access to specialists, this guideline could really just kind of help bring everyone to the same level of care.
And certainly the primary care provider can really sort of use these to say, we're best to make some referrals and really help that process. But again, that primary care provider will have no understanding of really some of the issues we raise in these guidelines and things that need to be monitored to really improve the quality of life of the child with Sanfilippo and even the adult with Sanfilippo.
Yeah. Well, as part of this process, you highlighted that a lot of what this is, it's guidance. It's certainly not rules or a regimented approach, but we did want to be as scientific as possible, given what was available in the literature and the studies that have been done so far. So the process matters of how guidelines are developed. But as part of this, much of it we relied on expert opinion. Now, to mitigate that, opinion and any bias that came from opinion is we really tried to cast a wide net of experts out there around the world. So to bring in perspectives from different geographic locations, countries, cultures, and languages and healthcare systems. In this process, over 100 people were involved in developing these guidelines. So I feel really proud that we really had a big net of outreach on that. I'll just take you quickly through the process. We had our steering committee formed, which included our 2 patient advocacy groups and an international group of clinicians, moved on to literature review, and then identified additional group of guideline developers who were also experts in their respective spaces in seeing Sanfilippo patients. Ultimately, we ended with 185 draft statements, which were then put out to survey to another large net of people that see patients with Sanfilippo across 21 specialties, 14 countries, and we really had a nice group, a large percentage that had seen at least 10 Sanfilippo patients or more, which is quite a lot given this rare disease. Through that process, we moved on to round 2, again, narrowing ones that met consensus, which was deemed to be a 75% response rate of either strongly agree or agree on the survey. So round 2, we had less statements to then seek consensus on, ultimately leading to our final group of recommendations, which were 178 statements. That then the steering committee worked to prepare into a manuscript.
And one of the keys is that even though 178 seems like a lot of statements, we have some nice tables in the publication that really help people focus on some of the key issues.
Yep, I agree. One way that we can look into get a little glimpse into the guidelines is by thinking about a clinical question that might come up and how they might be used, practically speaking. A family in a Sanfilippo support group shared this question recently about when they should be asking about orthopedics care for their child. And the clinical scenario is this was a 7-year-old child who had been seeing physical therapy and was wearing braces on their ankles for support and for some other things, but hadn't yet seen an orthopedic specialist. So when we look into the guidelines table, Dr. Muenzer, maybe you can walk us through this.
Yeah, so what this is sort of give an example of just one aspect of the disease process in the Sanfilippo. This is just looking at some of the orthopedic issues. And so our diagnosis clearly, we know there's issues with particular aspects of the bones, particularly scoliosis, curvature of the back, and a unique feature for Sanfilippo is that they could have bilateral hip disease, and so, or even hip disease that have to always be bilateral. So there's some basic things we would recommend just to get a sense. And then on a regular basis, what we'd like to see, typically the primary care provider, doesn't have to be an orthopedic person, is to just follow along and try to assess. For example, if you're concerned about the scoliosis getting worse, maybe x-rays are helpful. That clearly should prompt a referral at some point to orthopedics because we can do something about severe scoliosis. And so it's just worth sort of doing things on a regular basis. But this is where the guidelines can help the primary care, but that is something should really trigger a referral to orthopedics. The patient we talked about, if there's really a concern, it may be helpful to get a second opinion with orthopedics, or if you see progression of disease. We'll talk about in a couple slides on, if there's significant issues of pain or discomfort, that could be due to orthopedic issues, so that should prompt a referral. These days, we know that vitamin D levels are really low in a lot of individuals, and vitamin D has a huge impact in a lot of different aspects of the disease, which we won't talk about. But so, to monitor for that in terms of getting serum vitamin D. And then for the patients, unfortunately, with Sanfilippo who can become very immobile, there can be an increased fracture risk with that immobility. And if people are concerned, then a bone mineral density can sort of judge how significant that risk is and what you can do to sometimes mitigate that. So this just shows you that here's an example of not everybody needs to see orthopedics, but again, depending on what you find, may be needed to just help along along, help the primary care provider and the geneticist monitor the disease progression.
And, speaking to some of the baseline studies and where they can be valuable, sometimes we think, well, is it necessary if they might not be having an issue with that particular body part at the moment? And I think as things change down the road, if we don't necessarily have an understanding of where things were before maybe the child started developing pain or a symptom, sometimes it gets a little, it is difficult to tease things out for children with Sanfilippo who are not able to verbalize or localize discomfort. And so sometimes having these baseline understanding of what their normal is can be helpful in that process down the road.
Well, absolutely, Cara. I think that's an important consideration because disease can change. And if, say, 5 years down the road, you have an issue, but you don't have any exam for 5 years, it makes a big difference whether it's in the last few months or it's been there for 5 years, how you proceed with evaluation and management. So even though it's difficult for families to get seen, it's hard to get x-rays, sometimes they can be extremely valuable to try to get a sense for what the progression of the disease and when to intervene in the future and how significant it is.
Yeah. Well, let's go on to another clinical scenario that I think probably every parent with Sanfilippo and doctor that sees these kids regularly can identify with. And it's the help, my child's been yelling and in distress and something's wrong. And I tried every kind of thing in my checkbox list as a parent to figure out what that is. And I don't know. And I'm kind of at my wit's end because it's horrible to see your child knowing that something's upsetting them. And I imagine it's hard for a clinician too, right? In that scenario.
Yeah, as you allude to, this is one of the most challenging aspects of the Sanfilippo population is they can have these periods of irritability, they're uncomfortable, they're just not sleeping, and it's just not clear what it is. And so if you go to the next slide, this issue of, is some of the key evaluations this guidelines come up with for patients in pain, distress, or significant behavioral changes that were not clear of the etiology. And I won't go through all this list, but it's just that do easily, like not easily sometimes, but just we can get lab work. But I've had children who've needed to have a anesthesia event to really examine them thoroughly to make sure there was nothing going on. That'd be like, sometimes they just were so difficult and so uncomfortable. But one can't ignore that, this could be due to, brain issues. The irritability can be due to, ear infections. So sometimes they're unexplained infections, Typically, it's not eye pain, but evaluation of the eyes can occur. Dental is a huge issue. I've seen more than one child who've had a dental abscess, and that was the cause of their irritability. But if you don't think about it, and so this list is here just to get a sense for what the caretaker should think about as part of the process to try to figure out. I can tell you that we don't always figure out what happens. I mean, sometimes it goes away on its own for reasons I don't understand. We know these patients typically have significant neurological issues, they're cognitively delayed, and why it comes and goes. But sometimes you can identify clear-cut explanations, and that's important, like an infection. And so sometimes the labs was the first place I start is to look for any evidence, is there an infection going on, because that's something we can treat, But this is just guidelines to help people because this is a very challenging issue. You're not going to always get resolution, but at least you're going to rule out some of the obvious treatable things that we can deal with in medicine. Yeah,
absolutely. This concept of, not writing it off as, oh, this is just, these things that you're seeing with your child in distress, that's just part of the disease. No, we're going to kind of work through an individualized appropriate algorithm or clinical scenario, keeping these things in mind to make sure that we've not missed something that we can address. And like you said, sometimes we go down the list and we're left with, we haven't found an identifiable cause. One thing that we see in the Sanfilippo kind of parent community is we certainly recognize that these children, when they have discomfort of some nature, and I'll use maybe a GI disturbance, or if they're having constipation, or they're having diarrhea or reflux, is that anything like that seems to really set off the neurobehavioral symptoms that may have kind of been smoldering, baseline manageable, but become really over the top when they have some other layer of discomfort going on.
Absolutely. And I think one of the other keys for me is that sort of how quickly are these things occurring? If you sort of notice these within a few days where things get really bad, you're doing well and then all of a sudden things really fall apart, you're more likely to find something. Where if it's just been slowly going on, getting worse and worse over months, our chance of finding something is much less likely. We still need to look because, again, we can improve the quality of life sometimes recognizing things, but it's just important to sort of get a sense. And that's where, just having these guidelines, when you talk to a physician, you can say, well, maybe it's dental or this or that, but it's important to realize that there's so many different aspects that can result in pain, discomfort, and abnormal behaviors that one needs to think about.
Mm-hmm. Yeah, and as families look through this list and will have access to the same resource, they can begin thinking through and thinking back into the history and the things that have surrounded this event so that they can be better history givers and better participants in the child's care team. So I think all around, hopefully, this will help children feel better faster if it's possible.
And we also appreciate there's a lot of the Sanfilippo children that I think have a relatively high pain tolerance. So that's the other message, that you shouldn't ignore something that you think, oh, well, that's not too discomforting in a child. But when they start complaining, a lot of them do have a significant pain tolerance. And so it's important to realize that there may be something going on, even though, for some non-Sanfilippo physicians, oh, that's not a big deal.
That's a great point.
It's just, that's where these guidelines are helpful for people need to look. Again, you're not going to find sometimes what you want to find, but you're going to at least have a better chance to rule out the treatable causes of abnormal behavior, pain, discomfort.
Mm-hmm. Which I think is comforting in a way, to families and the doctors, that we've not missed something. At least maybe you're not making it better, but you are making it better in the sense that you've done what needed to be done. Yeah.
And our hope in the future, as we can treat the brain disease, we really won't be dealing with these issues and these behavioral issues, which really are probably exacerbated by pain in other ways, will be an issue, not be an issue, and we can really deal with just, helping the child manage the everyday things we see, like, dental issues are an issue for any child.
Yeah, absolutely. So in summary, our next steps for this project are that the manuscript is currently under review at journal for hopeful publication, and they're looking at it with their experts, so we're glad to be at this stage. In addition, there will be development of user-friendly format of this guidelines for families and providers so that it can be kind of portable and easy to use, and also be translated into multiple languages to increase access to this information. We also know that this is just the beginning, and future updates will be needed as care changes, as treatments emerge, and hopefully we see a change in the disease trajectory over time. I want to thank everyone again who participated in this project, our steering committee, guideline development group, survey participants, our advocacy partners in Australia, and Global Genes and BioMarin, who made contributions to help make this work possible. If you have any questions, please contact us at contact@curesanfilippofoundation.org. Thanks so much. Thank you, Dr. Muenzer.
Thank you.
Hello. Heading right into our next session, research happening now, targeting alternative disease mechanisms and secondary storage in Sanfilippo. We are excited to have two more amazing scientists with us here today. Dr. Alessandro Fraldi, Associate Professor of Medical Genetics, University of Naples, Federico II, and Principal Investigator at CEINGE Institute in Naples, Italy. And he'll be followed by Dr. Alexey Pshezhetsky, Professor of Pediatrics and Biochemistry at the University of Montreal, Canada. The stage is yours, Dr. Fraldi.
Hi, thank you, Cara. Thank you for organizing this very interesting meeting. So the topic of my talk today is the interplay between protein aggregation and dysfunction of autophagy lysosomal pathway in Sanfilippo and the related disease, and its implication for the therapeutic for the therapy of this disorder. Next slide. So, very brief introduction on mucopolysaccharidosis. As you know, these are inherited disease belonging to the large family of lysosomal storage disease. There are 7 types of MPS, each caused by the deficiency of a lysosomal hydrolysis required for the degradation of GAGs, which consequently accumulate as primary storage into the lysosome. Next slide, please. Neuropathy is the most important clinical feature in MPS, including Sanfilippo syndrome. And in fact, neuropathy is the major therapeutic target for this disorder. Next. However, today, there is still an unmet medical need in clinical care of MPS patients. In fact, currently there are no therapeutic options that can treat effectively the neurological manifestation in MPS patients. Indeed, next slide, please. Indeed, most of the therapeutic approaches that are currently exploited in treatment of Sanfilippo syndrome and in general in many mucopolysaccharidosis are based on correction of the primary defect, so the enzyme defect of GAG storage, for instance, by using gene therapy or enzyme replacement therapy. However, the clinical translation of this approach is highly challenging, and this is because the difficulty in achieving and maintaining the therapeutic threshold level of the enzyme into the brain before irreversible neuropathogenic cascade take place, and avoiding the use of high dosage of vector or the enzyme that could be potentially toxic or even costly. So basically, addressing this issue, of course, is important to improve the clinical translation of this therapeutic approach, and a way to address this issue, of course, is trying to target secondary pathogenic pathway downstream the primary defect, so trying to inhibit these secondary pathogenic pathways. Of course, next slide, please. Of course, to target pathogenic pathways, it's important to understand the mechanism underlying. So some years ago, we got interested in protein aggregation. We know that misfolded protein normally are degraded. However, when the degradation is impaired or reduced, basically misfolded protein can aggregate, and sometimes can aggregate in amyloid-like fashion. There are many proteins that can aggregate in amyloid, such as alpha-synuclein, A-beta protein, prion protein, tau protein. So these are very well-known proteins because they are hallmark of protein aggregate in many neurodegenerative disease. Next slide. So why we got interested in protein aggregation in MPS? First, as I mentioned before, amyloid aggregation is an hallmark of several neurodegenerative disease. characterized by lysosomal dysfunction, such as late-onset neurodegenerative disease, Parkinson or Alzheimer's disease, but also other neurodegenerative disease. Amyloid protein aggregation has already been observed in several neuronopathic MPS, including the brain of post-mortem patients. However, the relevance of this aggregation for the neuropathological progression has been so far poorly explored. Next slide. So to address the relevance of protein aggregation in MPS, we used a very well characterized mouse model for MPS IIIA. This is a spontaneous mouse model in which we can observe an early stage of disease progression, intermediate stage, and a late stage that mimic very well the progression of disease in patients. Next slide. So by using theoflavin staining, we found, as expected, a progressive aggregation of amyloid protein to the brain. As you can see, theoflavin staining marked several amyloid protein. Next slide, please. And we found that alpha-synuclein was the main component of this amyloid deposit, as you can see here by immunohistochemistry in MPS III brain and Western blot analysis with alpha-synuclein antibody. Next slide. And by colocalization analysis of alpha-synuclein and theoflavin, so showing that indeed alpha-synuclein is a component of theoflavin positive amyloid aggregate. Next slide. However, the amyloid deposit also contains other amyloid protein. For instance, we found prion protein, tau protein, and beta peptide. Next slide. So there was a progressive accumulation of multiple amyloid protein into the brain. So to address if this aggregation was relevant for neuropathology, we used a class of small molecule called molecular tweezers. These are potent and broad spectrum inhibitors of protein aggregation. These molecules inhibit the self-assembly of amyloidogenic protein, act on different amyloidogenic protein. So these molecules are not specific for a amyloid protein, and act by a unique process-specific mechanism involving the binding of molecular tweezer to lysine in misfolded protein. And this binding is moderate or low binding, and is also characterized by an high rate of on-off. So basically, this type of binding does not interfere with normal physiological processes. So this is specific for the self-assembly of amyloidogenic protein. Molecular tweezers rapidly concentrate the lysosome, have effect in vitro and in vivo on toxicity associated to aggregation in different models of neurodegeneration. This has been already demonstrated, and side effect can be observed at very high concentration. And then the molecular tweezer can cross the blood-brain barrier. CLEAR1 is the lead compound, the molecular tweezer. Next slide. And we used CLEAR1 in MPS IIIA mice to prevent amyloid aggregation. So basically treated systemically and continuously the mice with starting at 4 months of age, so before massive aggregation took place, to prevent amyloid aggregation, and these are the results. So basically we observed a strong reduction in amyloid aggregation, as you can see by theoflavin staining and immunohistochemistry against different amyloid protein. And this reduction in amyloid aggregation that we expected, next slide, was associated, this most importantly, associated to the amelioration of neurological signs, as you can see by neuroinflammatory, by reduction in neuroinflammatory marker in the brain, and also by the contextual field conditioning test, in which the mice treated with the drug recovered a normal behavior. Next slide. most importantly, what that we found by mechanistic point of view is that the neuroprotective effect of a CLEAR1 in MPS mice was mediated by the relief of the autophagy lysosomal pathway. So we know that in Sanfilippo syndrome, but in general in several mucopolysaccharidosis, the autophagy pathway is blocked. So the autophagy flux is blocked at the stage of autophagosome maturation, and this is very important for the neurodegeneration in MPS. And next slide. So what that we found when we used the CLEAR1 is that CLEAR1 normalized the lysosomal enlargement and reactivate autophagy flux. So this is MPS mouse brain, in which you can see a severe enlargement of lysosomal compartment. Next slide. That was associated to an accumulation of autophagosome into the brain, and also to accumulation of another important marker of autophagy flux, that is ubiquitin Ki63. Next slide. And when we treated mice with the CLEAR1, basically we observed a normalization of lysosomal compartment associated to a reduction of LC3 staining, thus supporting the conclusion that CLEAR1 was able to reactivate autophagy flux, as also showed by Western blot analysis against P62 and LC3 that are very well known marker of autophagy. Next slide. So as I mentioned before, in MPS, autophagy is blocked. We observed that the CLEAR1, so the inhibition of protein aggregation, was able to reactivate autophagy. However, we asked what is the mechanism underlying. So how amyloid affect autophagy flux, basically. This is an issue that we are addressing now. And I will show in the next slide some unpublished data. So what did we found that amyloid deposit accumulates in close proximity to the lysosomal compartment in MPS mouse brain. As you can see here, this is the colocalization lamp by confocal super resolution with the theoflavine. And this is confocal image showing the colocalization of alpha-synuclein with the LAMP1. Next slide. So the colocalization of amyloid to lysosomal compartment leads to a clustering of lysosome in the perinuclear region of the MPS mice. Here, in fact, what that we observed in MPS 3A mouse brain untreated. So basically, on top image, you can see the clustering lysosome in the perinuclear region. In bottom image, you can see the LC3 that indeed accumulate in the peripheral region of the brain. And in fact, next slide, LC3 and the LAMP1 do not localize. And LC3, as you can see by the bottom image, colocalized with the neurofilament H, that is a marker of axonal projection. So basically, this meaning that in MPS 3 mouse brain, last lysosome clusterized in perinuclear region, while LC3 and autophagosome instead accumulate in the peripheral region. When we treated mice with the CLEAR1, next slide, basically what we observed is that lysosome recovered normal positioning. So the words as reduced enlargement of lysosome associated to a normalization of lysosomal position, and this was associated to a reduction in the number of LC3 in the peripheral region. Next slide. So basically, the working model that we are proposing, that in a healthy condition, lysosomes are functional and can encounter and fuse with the autophagosome that are continuously formed in the axonal projection and travel along the axon to fuse with the lysosome. Instead, next slide, when in MPS condition, amyloid accumulates and aggregates in close proximity to the lysosome, and this caused in some way, by mechanisms that still are unknown, cause clustering of lysosome and an enlargement of lysosome in the pericardia. Next slide. As consequence, lysosome are less cannot more or better are less available to encounter a fuse with the autophagosome that consequently accumulate in the peripheral region of the neuron, and this at the end cause impairment of autophagy flux. Next slide. And impaired autophagy flux in turn can boost amyloid deposition by generating a vicious cycle. Next slide. So we speculated that the GAG storage, the primary GAG storage, can trigger in some way the amyloid deposition, and this because it is known that GAGs can form a scaffold to which protein, amyloid protein, can bind and aggregate. However, next slide, as I mentioned before, we still don't know how amyloid can cluster a lysosome and causing enlargement of a lysosome and trafficking problem, basically. This is something that we are addressing now. Next slide. So basically what that we can conclude is that molecular tweezer, and specifically CLEAR1, that is the lead compound of molecular tweezer, can be used as therapeutic, as a new therapeutic approach to improve the and to protect against neurodegeneration in Sanfilippo and likely also in other neuronopathic NPS. However, what we are proposing, next slide, what that we are proposing is a combined approach. So basically we are proposing to combine molecular tweezer to inhibit the secondary amyloid storage with the gene therapy to correct the primary defects. So why we want to address this approach? Basically because we believe that by this combined approach, we can improve the therapeutic efficacy and enhance the clinical translation of the therapy, basically for two reasons. The first reason, next slide, is that molecular tweezer may synergize with gene therapy by relieving residual amyloid burden, which may persist after gene therapy is applied, thus potentially reducing the dose of viral vector required to achieve the efficacy. And then because combined use of molecular tweezer may also extend the intervention window for gene therapy by keeping amyloid from building up until the full effect of gene therapy takes place. In fact, we have also some preliminary findings showing that in NPS mice treated with the gene therapy, there is still an amyloid burden. So this supporting the concept that using molecular tweezer can, in combination with gene therapy, can improve the therapeutic efficacy. So this project is, next slide, is supported by Cure Sanfilippo and Sanfilippo Children's Foundation that are supporting us in providing the proof of concept for this combined approach. Then I would like to thank also the other sponsor, Teleton and the Orphan Disease Center, and of course my lab and our collaborator at TGen, and Gal Beaton at UCLA, and Thomas Schroeder at the University of Essen in Germany, who is the person that generates the molecular tweezer. So thank you very much.
Thank you. Thank you both for the really interesting approaches and the detail you provided. I think they're both exciting up-and-coming things to translate in Sanfilippo. We have a couple of questions. For Dr. Fraldi, is the collective amyloid protein aggregation in Sanfilippo considered a damp, similar to GAGs and gangliosides, and therefore inflammatory?
Okay, so Basically, it's likely that amyloid storage can some way trigger inflammation. This is something that already know that amyloid can induce inflammation. So we believe that blocking aggregation, in fact, amyloid aggregation, can not only restore a normal and efficient autophagy flux, but it can also restore inflammation.
Thank you. Dr. Fraldi, have you investigated the lifespan of the MPS IIIA mice treated with CLR01 and looked at other behavioral aspects of that treatment?
No, not at all. About the survival, we didn't test the survival of mice treated with the anti-amyloid. It's something that could be done. Regarding other tests, yes, the answer is we also tested mice in other behavioral tests, such as open field, for example, and we observed also a rescue in the behavioral also in this test.
Thank you. Okay. Someone brings up the topic of, amyloid and neurodegeneration, and, thinking about the challenges that have happened in the adult neurodegeneration field with amyloid as a target, and, whether those are because of the treatments that were applied or other reasons. But, maybe you could speak to it. How is Sanfilippo maybe different or more suited for this approach?
So this will require maybe another meeting. However, I think that also, just to be brief, basically there are many similarities, and it is well known, between Sanfilippo, lysosomal disease in general, and late onset neurodegenerative disease. So basically, they are both characterized by lysosomal dysfunction and aggregation of protein. So they are both characterized. The problem is that, the difference is that what come first and when. So basically, in lysosomal disease, lysosomal storage disease are the paradigm of lysosomal disease with amyloid aggregation. So basically, lysosome become dysfunctional because there is inherited defect in lysosomal enzyme. And so this causes also a early aggregation of protein compared to the late onset neurodegenerative disease, in which the aggregation occurred only at the late stage of the life, of course. And of course, what that we learn by treating the late onset disease can be very useful for the treatment of Sanfilippo and in general for lysosomal disease. So I think that this is the first time that an anti-amyloid drug is tested in a lysosomal storage disease.
Mm-hmm.
So
yeah, I think we have to think about it as, this is a different scenario while there are those similarities in end product. The process there is quite different.
Exactly.
Yeah. Okay. I think it's so important, certainly the work that everyone's doing, but also these approaches that kind of, we need to go with the problem in all directions and build our toolbox for children and doctors so that we have interventions, that may be better suited to one patient than another at different points in their lives. So that we are continue to hope that regulatory agencies will use flexibility with such a devastating condition. And thank you both so much for your work. We will wrap up this session. welcome back everyone to our next session titled Funding Opportunities for Research in Sanfilippo. And we're grateful to have with us today Dr. Jill Morris from the National Institutes of Health, where she serves as Program Director in the Division of Neuroscience at NINDS. And following Dr. Morris, you'll hear a little bit from me about funding opportunities at Cure Sanfilippo Foundation. But first, welcome Dr. Morris. You have the stage.
So thank you very much for having me. I've been listening to the meeting and I have very much enjoyed it. In particular, it's very helpful to hear from the family members and to get their perspective and the challenges that they face. And it makes me start thinking about things that NIH can do to try to help out. So as Cara said, I'm a program director in the Division of Neuroscience at NINDS. I'm in the extramural portion of NIH, which is responsible for the management of funding of all the grants across the country. So we represent all the academic and business research facilities across the country. And then intramural at NIH is the actual clinical center, and it's like its own academic organization. Next slide. So Walter Koroshetz is the director of NINDS, and the mission of NINDS is to seek the fundamental knowledge about the brain and nervous system and to use that knowledge to reduce the burden of neurological disease. We have a website. I encourage you to go to it. Walter has director messages that come out every few weeks, and we also do press releases on new discoveries that have been made by an NINDS funded investigator or new funding opportunities that come out. Next slide. So NIH in general has numerous funding mechanisms. The vast majority in the 90%, I don't know if it's 95%, somewhere around there, of grants that come in and are funded are through the investigator-initiated R awards. So you often hear investigators talk about R01, R021, or R03 award. And these are the awards that typically will fund a lab and maintain its research over the long term. But NINDS also has specific funding opportunities related to translational research, as well as clinical research. Next slide. So I'm going to talk today about a few funding opportunities we have, or that we have coming out, like the one shown on this slide, that focus on ultra-rare disease, rare and ultra-rare disease. So we have a funding opportunity that's been cleared by council and will be released in the future, near future. And it's focused on the identification of clinical outcome assessments for ultra-rare neurological and neuromuscular diseases. So the goal of this funding opportunity will be to identify and evaluate the utility of clinical outcome assessments that will be used to determine efficacy in clinical trials of candidate therapeutics for ultra-rare neurological and neuromuscular diseases. And when we looked over the landscape of what we had available, so in red, we have numerous programs in our translational group in order to develop therapeutic modalities to move forward into the clinic. We also have numerous funding opportunities to support clinical trials. But in particularly in the ultra-rare disease space, getting funding to do a natural history study, we've heard from others in the community that this is particularly difficult. So we are working on developing this funding opportunity so that when you have a therapeutic modality, you'll be able to use it, and then in the when the when the clinical trial occurs, you'll be able to determine if there's efficacy. So, for example, in cognitive assessments, yesterday when people were talking about the difficulties with sleep, one can imagine if there was patient-reported outcomes from the caretaker that sleep had improved. That would be an example of an outcome assessment. Next slide. So for this, we're in particular focused on the ultra-rare space. And so the US doesn't have a specific definition for ultra-rare. We do have it for rare disorders, but we're using the European definition, which is 1 in 50,000 persons, which is approximately having fewer than 6,500 individuals in the US at one time that are affected. And certainly the Sanfilippos fit into that category. The initiative will allow for retrospective and prospective natural history studies. We would like to see innovative and efficient study design. So for example, remote assessment, because of the limited number of patients, it may be difficult for one particular institution to go to see all the patients across the country or the world to assess them. The community should be involved. So again, hearing the patient perspective gives the investigators a feel for what they should actually be looking at and what really affects the quality of life for the patient. We are encouraging the use of existing infrastructures, collaborations and resources. Typical for NIH, we require the use of rigorous and scientifically appropriate statistical methodologies. So, and we realize that when it comes to a very small patient population, the statistics will be difficult, and that, we completely understand. And there will be a requirement that there's a need there, that there are therapies in development that may be within the next 5 years. Next slide. We already have a clinical trial readiness for rare neurological and neuromuscular disease that focuses on validation. So this says after you do the natural history study, you have a clinical outcome assessment or a biomarker, and you want to validate it across multiple sites. This may be more in the rare disease space where there's more patients, but we do also have this funding opportunity available. Next slide. And then finally, I just wanted to note, we have a general biomarkers discovery program. So we have, this is for any, rare or common disorders. So we range from, biomarker discovery all the way to the validation state. So that's available as well. Next slide. So I just wanted to touch on a few of our translational opportunities. So we have a division of translational research, and the mission is to accelerate basic research findings towards patients' use for neurological disorders and stroke by providing funding, expertise, and resources to the research community. And we have the whole spectrum from early assay development and screening to early clinical development and going into clinical trials. Next slide. So I really encourage you, if you're interested in any of these, because I'm going to just briefly touch on them, but we have reoccurring webinars on each of the funding opportunities, so you can learn about them. And then we actually included in those webinars are just general webinars about other NINDS funding opportunities that not necessarily translational that you may be interested in. So if you go to this website, or this is how I do it, I Google NINDS Division of Translational Research, you'll come to this general page entitled Translational Research. You scroll down to the bottom and there's a tab for resources and tools, and then you click on Join Our Listserv, and then you'll get reoccurring emails that will tell you when these webinars are going to be taking place. Next slide. So I wanted to mention our early translational funding opportunities. So we have 3. This is called the IGNITE program. And one is for the characterization of a therapeutic agent. And then these are phased awards. So it's a 2-phased award. So the first phase, you would identify, you would characterize a therapeutic agent. And then when you reach a certain milestone, you would then go on to prove its efficacy or validate its efficacy in an animal model. We have one for assay development and therapeutic agent identification. So again, the first phase would be the development of the assay. And then the second phase may be a small screen to test, to screen for our initial set of compounds. And then we have one for improving model systems in order to show that you're engaging a target. So for example, the easiest one to describe is in tumor models to add some type of luciferase or enzymatic reaction that you can show and visualize under imaging to show that you're engaging the target that you want to. And these projects are for 3 years, and the total direct costs over the 3 years is $750,000. So typically people come in at $250,000 per year. Next slide. So I just put this slide up because this is true for all of our translational programs. So this is the example for the IGNITE. But we have other, I want to talk about our urgent program, and they're all milestone based. And the reason that we do milestones is, especially in translation, when these grants get reviewed by our review committees, people will come in with some high-risk projects. So they'll suggest developing a very innovative assay that may or may not work. But if it does work, it could be a game changer for the field. And so when reviewers see something that's really high risk, they get nervous about funding something for the whole term of the award. Whereas if there's a milestone in place that even though it's risky, but if it doesn't work out, the award will end, that gives reviewers that reassurance that there's an endpoint for those riskier projects. So even though it makes investigators nervous that they have to reach a milestone, in the end, it's good for the review process because you can come in with more innovative, high-risk projects, and it gives those reviewers some comfort to know that if it doesn't work, if it completely fails, there's an endpoint. Next slide. So one of our newer programs that came out this last year is the Urgent program, so Ultra-Rare Gene-Based Therapy Network. And this program is designed to provide the opportunities for groups to come in and be able to be provided with resources. So this is specific for ultra-rare disease. So again, it's the qualification of have to qualify for the definition of ultra-rare disease. The typical application is what we call the UO1. And an individual would come in and let's say they have proof of concept data. They, had early conversations with the FDA about what else they need to do. And they would come in and they would be the source to do all the biological assays that may need to be done in the mouse model or whatever model they have of the disease. But then we would provide contracts to do the pharmacology to manufacture the product. So this is all biology based. So this could be oligonucleotides, so ASOs, antisense oligonucleotides, or AAV viral vector, or an antibody. So any type of biological project, we would pay with a contractor the manufacturing, which can be significant, as you know, especially for manufacturing of viral vectors for doing clinical trials. And then we'd also pay for, through a contract, the tox studies. And what this provides the community with is they don't necessarily have to have the expertise in doing tox and doing pharmacology and doing manufacturing. They provide the expertise in the disease and the disease models. And so when a person comes in, this is a U award, these are cooperative agreements. We would put together a team in which the PI would be a team lead, as well as we have a group of subject matter consultants that would also be a team lead, and we would meet frequently as we move the product into development. And then after completion of all the studies and you get an IND, it would go directly into a clinical trial without having to submit another grant. We also have what's called an EXO-1, which is just providing the resource. You can apply just to have the vector manufactured or the ASO manufactured, or to have the tox studies done. So we wanted to provide a suite of options available to the investigator to move these therapeutics into the clinic. Next slide. So I also just wanted to briefly mention our small business program, and I was looking at the slide right before, and I'm not sure that so small businesses congressionally mandate set aside money. I'm not sure if it's still 3.65%, but it's totally separate from the academic research fund. So there's no competition between academic researchers and the small business program. It's a completely different set of funds that are mandated by Congress. And there has to be a potential for commercialization. I know Jill Wood is talking later, and she is an investigator who's been very successful at getting small business funding. But there are, the majority of the applicants are investigator initiated and come through our general funding opportunity. And we also have specific, very focused funding opportunities available through the small business programs. Next slide. And finally, I just wanted to, there's one after this, so almost finally, I just wanted to mention the Rare Disease Clinical Research Network. So NINDS is a major player with the RDCRN, which is run through NCATS. So this slide shows the 20 consortia that are currently funded. And NINDS is co-funding 11 of the 20, and we're also co-funding the Data Management Coordinating Center. And as some of you may know, that there is currently the Lysosome Storage Disorders Network. Because we put in a stipulation that they can only, any consortium only can have 15 years of funding or 3 cycles, that consortia will be ending at the end of this funding period. However, Sanfilippo's wasn't a major focus of that. So the Sanfilippos could come in as their own RDCRN and put together their own consortia. Next slide. So NCATS recently released a notice of an intent to publish. So the next round of competition to develop a consortia will be the earliest application due date will be December 2024. And so as a group of disorders, the Sanfilippos could come in as their own consortia and apply to the RDCRN. So I just wanted to make you aware of that. Next slide. And then just finally, we have other programs that are similarly designed to Urgent, where we provide you with a suite of resources, contractors that will do things like tox studies and manufacturing. One for biologics, I'd say there's one part of the biologics that's an advantage with compared to the Urgent is you can come in to do earlier stage proof of concept studies. So if you're you have more work that needs to be done to optimize your gene therapy, for example, you could come into that early stage of the Blueprint Biologics program. There's a small molecule program, and then just recently there's a device program that just came out called the MedTech program. Next slide. So I just want to thank you for your attention, and I provided the slide to Cara, and you can have access to them as well in the future. But thank you very much.
Thank you, Dr. Morris. Some really amazing opportunities at NIH, and exciting to hear about future opportunities with the RDCRN as well. I encourage any of the researchers listening to follow up with Dr. Morris at her email if you have questions about any of those programs. And of course, we would love to be collaborators on any of these programs moving forward. And certainly we've talked about many of these things over the years, Dr. Morris. So thank you for that. And next you'll hear, it's a little recording, but just sharing with everyone our foundation's funding opportunities coming up. I think one thing that disease-specific foundations like ours do, certainly we fund things across the gamut, including clinical trials, but we also serve a really important role in the early pilot stage projects that can build the data needed to apply for NIH funding. Yeah. So thank you, and
thank you.
Cue the recording. And collaborate on research ideas. Hello, and thank you for joining me again. I'm Cara O'Neill, Chief Science Officer at Sanfilippo Foundation. I'm here to talk to you about some of the funding opportunities that we have at Sanfilippo. One of the wonderful things that I get to do here is collaborate with scientists, clinicians, and many other folks around the world who are working in this disease, and collaborate on research ideas and the gaps that exist. Now, the impact that the foundation has been able to make over the last 9 years is really remarkable. And more than $10 million in Sanfilippo-specific research funding has been awarded in that time frame, thanks to incredibly generous donors. This has provided support for more than 35 research projects and multiple clinical trials. Our research funding cycle is about to open this July 11th, so Monday, in which letter of intents will begin to be accepted. We will move through the process, evaluating those letters of intent, which are an initial summary of the proposed research. We will look for alignment with our goals and research needs and the quality of the research proposed. Select applicants will be invited to submit full application, and the deadline for that will be on September 30th. All applications then go through a process of peer review. One unique thing we do at the foundation is provide that feedback, both internal and through external peer review, to applicants. So they have the opportunity to learn from that feedback, as well as respond to any questions or comments that arose, and have the opportunity to resubmit their application. After that point, the Scientific Advisory Board then reviews all the applications, and those that meet a funding level of merit will then go to the board of directors for review and funding decisions. We anticipate that applicants will be notified of these funding decisions in mid-January. Our typical funding mechanism at Sanfilippo has really been quite open, but we recognize the need to focus in on a couple of different areas, and so we have 2 new funding mechanisms this year. And I'll run through those briefly. We have a pilot funding mechanism where we're really looking for that early stage research or new ideas, and that's about $50,000 per year, which can be renewed up to 2 years. We have our advanced Sanfilippo funding mechanism, which is our traditional, rather open funding mechanism for those larger scope of work and funding need applications. And we've also established a fellowship award mechanism. This will be $60,000 per year for up to 2 years. This will be highly competitive and need-based. The goal and intent of this program is really to build this next generation and pipeline of talent among early career scientists and clinicians who would like to focus on Sanfilippo syndrome. We'd also like to be able to draw talent from related fields who can enter the space if given the support and further enrich what's going on in Sanfilippo research. Again, we always look at where the gaps are and what we think are priorities to fill that will help advance either clinical trials or quality of life. And some areas that we have recognized the need to focus on are around the gastrointestinal system, the peripheral nervous system, some more work around personalized medicine, gait and mobility, and movement disorders in Sanfilippo. Now, while applicants may submit research proposals on anything in Sanfilippo, and certainly we want to hear your incredible ideas and want to see that innovation, these are areas that the foundation has recognized as important to address. So where will you find this information? Go to our website, go to the How to Help, search down for researchers and funding opportunities. So check out the website on Monday, and please contact me if you have any questions, or contact research@sanfilippofoundation.org. Thanks so much. Welcome back. Thank you for joining this next session entitled Moving Towards the Clinic, Advancing Translational Programs. This is our second such session, and we are very happy to have with us today Dr. Anna Bakardjiev, Executive Director of Development at Denali Therapeutics, and she'll be followed by Dr. Tsui-Fen Chou, Professor of Biology and Biological Engineering at CalTech, along with Ms. Jill Wood, CEO and co-founder of Phoenix Nest Incorporated. Take it away, Dr. Bakardjiev.
Thank you, Cara and Glenn, for organizing this wonderful conference. I have very much to the presentations and hearing from families and other scientists and physicians, and also from the different organizations that are in this space. I also want to thank the Sanfilippo Foundation for inviting Denali to join this event and to share an update on our organization and our efforts to advance the treatment landscape for families living with Sanfilippo syndrome and other MPS disorders. Next slide, please. So I'm a physician and an employee at Denali. I will be talking about the drugs called DNL310 and DNL126. Neither one of these drugs is currently approved by any health authority. Next slide, please. And today I will provide an overview of our enzyme transport vehicle technology, or short abbreviated ETV. I want to share some learnings from our program in Hunter syndrome, or MPS II. I will show you interim 49-week data from our ongoing phase 1/2 study of ETV-IDS, or DNL310. And then I will give you an overview of our program in Sanfilippo syndrome type A, or MPS IIIA, with ETV-SGSH, or DNL126. So to make it easier, I will just refer to DNL310 and DNL126 in the next slides. So before delving into the science, I want to briefly introduce Denali. Denali Therapeutics was founded in 2015. We quickly built out our global capabilities, and we have now offices in the US, in San Francisco and Utah, and in Europe, in Zurich. We currently have 6 therapeutic programs in clinical studies, and we firmly believe in success through collaboration. Our therapeutic focus is on neurodegenerative diseases and lysosomal storage diseases, including Sanfilippo syndrome. And our approach is based on 3 pillars that we summarize under genetic pathway potential, engineering brain delivery, and biomarker-driven development. We believe that this scientific approach increases the likelihood of the success to develop effective brain-penetrant therapies for MPS and for other rare disorders. Our commitment is to listen to people and families living with LSDs. We engage individuals, families, caregivers, and advocacy groups in our work as we strive to develop impactful solutions that address your needs. Next slide, please. So treatments that cross the blood-brain barrier, treatments that cross the blood-brain barrier and aim to address the neurodevelopmental symptoms of MPS, such as developmental delay, disruptive behaviors, impaired cognition, remain a current care. And as you know, MPS is caused by mutations in genes that lead to a lack or low levels of enzymes that are necessary to break down long chains of sugar molecules called mucopolysaccharides. So it is difficult to replace these enzymes, especially in the brain, because large molecules like enzymes cannot cross the blood-brain barrier. So Denali has developed a platform that we call the ETV, or enzyme transport vehicle, where the enzyme that needs to be replaced, for example, in the case of MPS IIIA, it's the SGSH, or sulfamidase, or SGSH, or IDS in MPS II. These enzymes are bound to the ETV platform. And you can see that in the left depiction of the molecule. And then the ETV portion of the molecule binds the transferrin receptor, which is found on blood vessels in the brain and other tissues. And with the transferrin receptor, the ETV and the enzymes are moved across the blood-brain barrier. And this is a normal transferrin receptor is a normal receptor that is typically used for ion transport into the brain. And the ETV platform is designed so that it does not interfere with the normal function of the transferrin receptor. So next slide, please. ETVs are delivered by intravenous infusion, and they are optimized to cross the blood-brain barrier. And that meaning, in addition to not being designed to not interfere with the normal function of the transferrin receptor, they're also designed for optimal crossing of the blood-brain barrier, meaning that they are binding to the transferrin receptor, but not so strong that they're not released on the other side of the blood-brain barrier. Importantly, transferrin receptors are also expressed in the blood vessels of other tissues, and so they may also facilitate enzyme replacement in tissues such as bone, cartilage, and heart. And so ETVs have the potential to treat both the neurodevelopmental and the physical symptoms of the MPS disorders. So here's an outline of our program for Hunter syndrome, and also a schematic depiction of the DNL310 molecule. And I just briefly go over this program because the program is probably, the MPS IIIA program will probably follow a similar pathway. We have two ongoing clinical studies. The first one is an observational study in MPS II participants. This is a prospective multi-center, multi-regional observational study of participants of MPS II to assess biomarkers potentially related to disease severity and or treatment response, and to prospectively assess the progression of disease in children with MPS II. Then we have the ongoing phase 1/2 study of DNL310 in children with MPS II. This is an open label 24-week study to assess safety, pharmacokinetics, and pharmacodynamics of DNL310. We are enrolling 45 participants with MPS II with all phenotypes up to 18 years of age, treatment naive and on standard of care IDS. As you may know, MPS II has a standard of care enzyme replacement therapy that treats some of the peripheral symptoms. And we are enrolling into 5 different cohorts in North America and Europe. And the participant, after they have completed a 24-week study, they can enter an open label extension for continued evaluation. And then we have started recruiting for a phase 2/3 study. It's a double-blind randomized study of efficacy and safety of DNL310 and MPS II, and is currently recruiting in the US. And next slide, please. So on this slide, you can see some interim data from the phase 1/2 study. On the graph, you see the CSF-HS levels in up to 17 participants. And you can see that at baseline, all of the CSF-HS levels are elevated. And you can also see that the majority of participants across all cohorts and all dose levels normalized CSF-HS at the first lumbar puncture on DNL310, which was after only 4 or 6 doses of DNL310. Normalization of CSF-HS occurred in all participants at week 24 and was sustained at week 49. The weekly infusions of DNL310 were generally well tolerated at doses of 3 to 30 milligram per kilo. We have an independent safety and data monitoring committee that has reviewed all the data on a quarterly basis and has recommended continuing the study without any modifications. All participants reported TEAEs, which was not unexpected given the substantial underlying morbidity of individuals with MPS II and the duration of the DNL310 treatment. But the AEs, the adverse events, did not lead to any treatment withdrawal or study discontinuation. Infusion-related reactions were the most common treatment emergent adverse events, which is consistent with enzyme replacement therapies. So overall, we concluded that normalization of CSF-HS occurred in all participants by week 24. It was sustained by week 49, and weekly IV infusions of DNL310 were generally well tolerated. So on the next slide, here you can see a comparison of DNL310 and DNL126. which we are developing for MPS IIIA. So both molecules utilize the exact same ETV platform, optimized for brain delivery. There is only the difference in the enzyme that is attached to the platform. And for DNL126, we are actually attached 2 enzymes to each ETV molecule instead of 1. We also have 5 more programs in preclinical development, including a program for Sanfilippo Type B. So here you can see brain and CSF HS data in the MPS IIIA mouse model in mice treated with DNL126. So in both of these panels, you can see a bar with the black dots, and this represents the brain and CSF HS levels in an unaffected mouse. In the next bar that has the gray dots, you can see the brain and CSF HS levels in the Sanfilippo mouse model. And you can see that the HS levels are significantly elevated in both brain and CSF. And then the 2 bars with the orange dots represent the HS levels in MPS IIIA mice treated with either a single dose of either a low, high dose of DNL126. And you can see that even a single dose of DNL126 leads to significant reduction in brain and CSF HS, and that the high dose actually is very similar to the levels of HS in the control mouse. So we recognize this is only mouse data. However, these preclinical findings are important to determine if the investigational treatment may have an impact on the biomarkers of the disease in the clinic. So we are starting a natural history study for Sanfilippo syndrome type A. This study has the objective to gather data to better inform a future investigational treatment study in MPS IIIA. We recognize that there are studies that have been published, and we regret that we came to the decision that we need to do another study because we don't have access to all of the data, the domain-specific data especially, that has been conducted in the previous studies. However, we want to make it as easy as possible on the families and the participants. And so the study is designed to be done entirely remote in the participant's home. And we will do only 2 assessments. So we will assess adaptive behavior, which is the conceptual, practical, and social skills that the individual utilize in the daily activities, including communication, socialization, and personal care. And for this, we use the Vineland-3. A lot of you might be familiar with this. This is actually an assessment, it's a semi-structured interview of the caregiver, so not the participant or the affected participant. It's the caregiver, and then there is, in addition to the Vineland-3, we are collecting blood samples to assess blood-based biomarkers. We are partnering with this with Sanguine Biosciences, and I'm pleased to report that we just got IRB approval. And if you go to this link that is here in the slide, or go to our virtual booth, you can sign up to get more information if you're interested in potentially participating. Special thanks to the families and children that participate in our clinical studies. We very much thank the Cure Sanfilippo Foundation and Dr. Cara O'Neill for inviting us to join today and for your guidance and insights. We also thank our collaborators that are currently developing on the phase 1/2 study of DNL310, and we look forward to continuing to work with the Sanfilippo community as we advance our programs forward.
Hi, good afternoon. My name is Tsui-Fen. So I'm going to update you on our progress for ERT, the ERT development for MPS 3D. Next one, please. So this project will not be possible without Chou would to start a company, Phoenix Next. And this I list the team because, in my opinion, it's very important for the family, for the MD, and for our the researcher to work together to help develop treatment for this important community. And also, I list some of the paper here we published recently. And you have any questions, feel free to reach out. And special thanks to the NIH, Dr. Jill Morris and Emily, who actually support us, and we have monthly update meeting with them to help this project to move forward. Next one. So I don't need to explain to you what's the MPS 3D. I just want to say is that the enzyme is missing is called GNS, which is a sulfatase. And the biomarker we will use in our animal study is the GATE, heparan sulfate. Next one. And highlight again here, this is the enzyme that mixing in the type 3D subtype. Next one. So in this paper, we demonstrate proof of principle that we use MIC take GNS to show the efficacy in the neonatal mouse study, and also demonstrate how we can do the quickly proof of principle in the stable cell line with G41 select. But then later we use the methotrexate DH5 alpha selection system to improve the yield of the protein, which is important for ERT. We need to have functional and enough protein for the study. Next one. So, and this is the important study. We still use the MIC-TEC GNS, but we do the study in the adult mice. And this is a collaboration with Patty Dixon at WashU, and her technician Stephen inject the protein my postdoc Feng and San produced in the mouse. And the data you are seeing here, basically, we Steve cut the protein, the brain into 3 sections, and we check the enzymatic GNS activity to show with the administration of the purified GNS, the activity can increase dose dependently. Next one, please. Then importantly, we can see the brain, both brain and CSF heparan sulfate reduced to the carrier level. So this again, we can see in the dose dependent manner, we can see some efficacy at lower dose, but at 300 microgram dose, we can see complete reduction of the heparan sulfate in both brain and CSF. Next one. Then importantly, we have the heparan sulfate as biomarker to show the efficacy. But in my opinion, we need more good biomarker to help us to understand what is the disease mechanism. And the approach we choose to use is this unbiased proteomic approach, which you can see 10,000 of protein at one time. And the figure on the left is basically comparing the wild type mouse and the MPS 3D mice to see what are the protein that actually is changed caused by the lack of the functional GNS. Then the figure on the right, at the same time, we can give the treatment and use the same approach, look at 10,000 of protein at one time, and to see what other protein can be rescued. And this is a study still ongoing, and we want to see if we can apply this approach systematically at different subtype of MPS, and also using different sample from the mouse CSF and sample from the iPS-derived mini brain to see what is the systematic way to identify maybe new therapeutic target and also new biomarker for the clinical study. And we use similar approach for one of the anti-cancer agent development. And the protein we find actually now is in one of the clinical trial can be used to monitor the treatment efficacy. So now I will pass the baton to Jill. She will update you on the other side of this company is going on.
Hi, I'm assuming that everything's a go here and we can get started. Thank you, Dr. Chou. It was lovely seeing you and listening to your presentation. To echo that conversation, we are going to be discussing Phoenix Nest Program All 127, which is an observational study. We also have a few other programs, clinical programs, in our pipeline as well. We have the gene therapy program, which was presented by Dr. Sigmund Seti yesterday. We have our ERT program, which was just now presented by Dr. Chou. And if you guys got to catch Alexey's talk on our peptide molecule earlier today. Next slide, please. Again, as we discussed, we will be talking about all 127 today. And fortunately, we also had Jill Morris speak before me, and she gave us an overview of the National Institute of Health grants. And this project was funded by just one of those grants. Sri and I applied for this grant when it really became abundantly clear that we were not going to be able to really support our interventional drug intervention until we had sufficient natural history to back our claims. Our goals seem pretty simple, but when compared to the obstacles, we really do have our work cut out for us. Sanfilippo syndrome is very difficult to work with. It's not like SMA, where the babies can learn to walk after being treated with gene therapy. Our treatment effects are much more subtle. SAN-C and SAN-D are the rarest versions of Sanfilippo and have not had a treatment go to the clinic yet. However, the silver lining is that we have seen what the other trials have gone through and what has worked and what has not worked. Next slide, please. As part of one of our milestones, we took our protocol to the FDA. They had a lot to say and was actually really very informative, and I am greatly appreciative of all the feedback that we did receive. It might not have all been perfect, but we're going to be transparent here in hopes to help the other trials that have gone before us and will continue. So the assessments that we chose were basically based off of the other interventional trials. We used the VABs, the CABs, the Baileys. Yesterday, we heard from Tom Mathers of Alivix and Emil Kakis from Ultragenics speak on their two programs. As a little reminder to all of you, Alivix acquired their ERT program from BioMarin, and Ultragenics just recently acquired their 3A gene therapy program from Abeona. And I give you great applause. Thank you very much for picking up these programs and leading the way for Sanfilippo. Both Tom and Emil discussed endpoints and cognitive assessments used to measure their trial endpoints. They also referred to the change in the scores of their treated patients compared to the scores of the patients in their natural history studies. For the SAN, C, and D families out there, I want to remind you that there really is no natural history done for types C and D, whereas types A and B have been working on natural histories for over a decade now. The improvement of these scores have not been high enough to convince the FDA of meaningful change, and the FDA is asking these sponsors to continuing the youngest patients treated up to a certain age. And I think we heard Tom or Emil say it was to the age of 5. Unfortunately, time is not on our side. Next slide, please. So what can Phoenix Nest do differently or better to ensure a successful interventional trials for type C and D? As mentioned again, SAN, C, and D have very little natural history. We're uber rare. Our age range is all across the board, and our patients are spread around all across the world. So you have patients that are extremely high functioning, and we have patients that are at the end of the stages, but we have few patients that are right there in the middle. And the different languages and the translations is very difficult to work with when you're choosing cognitive assessments. And to top that off, our regulators have still not acknowledged the heparan sulfate is disease-causing and approved this as a reduction of heparan sulfate as a biomarker. So what are we going to do? I really went back to the table and immersed myself into the patient caregiver papers that have published. Again, credit to the Sanfilippo Foundation for taking the initiative to get these papers out there about Sanfilippo caregiver burden and the assessments that we would like to see and the improvements in our children that we would like to see. Not sure what, you know, as a parent, you'll ask any one of us, we don't expect our children to add and subtract. We don't expect our children to read. What we want for our children and for our families is quality of life. I don't understand why it is that we've had our trials have been using cognitive endpoints. Yesterday we heard the 3C parents talk about sleep and GI issues and how changes in these areas would improve the quality of their lives. As a parent, I completely agree with the importance of sleep for everyone in the family, and not having to clean feces off of public bathroom wall would make my life a lot better. But the question is, will our regulators approve the most expensive drugs in the world for improved sleep and a solid bowel movement? I don't know. Will our insurance companies cover these drugs? Again, without heparan sulfate as approved biomarker, the challenge remains. What endpoints can we use to verify meaningful change, and what are the clinical outcome assessments that we can use to measure the change? Next slide, please. Well, our team decided to focus on activities of daily living. And fortunately, the search of clinical assessments that reflected the needs of our Sanfilippo children and where they're at was extremely challenging. I'm pretty sure that I made the companies that hold the licenses to these assessments absolutely crazy. Like the person in the department store trying on every single shoe in the store. Nope, doesn't fit, doesn't fit. So exasperated, I turned to Jonas therapists, realizing that these are the people that know our kids best. I was really happy to see yesterday Suzanne give a talk that an ABA therapist, I hope you guys all saw that talk, which really echoes my experience in our children's needs, and that these guys can play an instrumental role in our children's lives, and also the development and the selection of clinical outcome assessments. So I can't thank Jonas' team enough. And here are some of the assessments they helped point me towards: the LIDER, the MFM32, the SFA, and the PLS5. And focusing on some of the areas in the caregiver burden, experiences and papers, communication, mobility, eating, socialization. And communication can be an expressive and what's my other word? Receptive language. Mobility, and mobility means different things to different people. And eating as well also means different things to different people as well. The ability of a child to use their fingers to eat, or the child using forks or spoons to eat, or the parent loading in the fork and feeding the child. And then the loss of not being able to chew on their own. Next slide, please. So to capture all these special nuances of Sanfilippo, we created an application, and that is exactly what it is an app that you download on your phone. The software was provided by Apparito. We worked with Casimir Trials to help us with the concept elicitation and task design. And together we came up with 10 activities that the families will record around the times that they have in clinic visits. And we have several questionnaires that they will also be answering throughout the study. The RARE app gives visual meaning to the clinical data, virtually showing the downward progression of the disease now and creating a baseline that we can use to prove stabilization of the syndrome or improvement in their ADLs after interventional treatment. Next slide, please. We're going for synaptic synergy here by layering the clinical data, the retrospective data, and real-time data together to paint the picture of Sanfilippo. To summarize and in closing, all 127 will take place at one site at Columbia Presbyterian with PIs Wendy Chung and Gustavo Miguel. We will be bringing all the Sanfilippo Type D patients that we know of from around the world to this one site. They will be followed over a course of 2 years, coming in 3 times annually. The trial will be a typical natural history style trial, all the same bells and whistles with the Sanfilippo twist, because it was designed with the insight of the parents and the caregivers and the physicians that really truly understand this disease. Next slide. And lastly, I would like to thank all the amazing people that have helped us on this project. Without the families, we would not be here today. A special thanks to the NIH for providing the funding for this study, and to Bernie Kuka, one of my new best friends, and our scientists and consultants, and to our CROs, LabCorp, Casimir, and Apparito. It really does take a village to make these clinical trials work. Thanks again for having me here, and conclude my talk.
Thank you.
Cara, you're on mute. You're on mute, Cara. You've been on mute since the beginning.
Sorry. Thank you all for the wonderful presentations. We'll start with the Q&A. Dr. Bakardjiev, we have a question for you. And looking back at your Hunter syndrome data, is anyone else getting this echo? I don't know. Okay. Wondering what you see as far as the baseline level of CSF heparan sulfate in Hunter syndrome versus, untreated baseline CSF in Sanfilippo syndrome. Are we starting from the same kind of point, or is, where does Sanfilippo fall in that?
Yes, that's a very good question. We think it's relatively similar. We have analyzed any CSF samples from Sanfilippo patients with our own methods, and one of the problems that in the literature methods used to these biomarkers. But when we review the literature, we think we find that in FHS levels are approximately about 10-fold higher than normal, and in Sanfilippo about maybe 13 or 14-fold higher. So it's like log orders differently, I would say.
Okay, thank you very much. Another question for you about the somatic effects of ETV, or maybe your entire product.
So we have very robust decline in not just the total GAGs that are colorimetric clinical assays, but we also have more sensitive assays where we use mass spec to measure and HS and we have a 80% or greater reduction in both urine HS and DS. So that suggests that we'll have we are improving the, and this is actually the reductions after we're switching from standard of care to DNL 310. So we improve the effect on the peripheral symptoms, but that will require, because we're already starting from the standard of care, it will require be able to say that whether we have a clinical improvement. And you know, some of the tissues that are not well treated, like the bone valve, they will probably take also a long time to show any effects. Not like liver and spleen, which, you know.
Sure. Yep. Thank you. Thank you for that. A question for the Phoenix Nest team, or maybe a comment about the observational study you're developing, and if you plan to apply that same strategy to your Sanfilippo C program.
Yes, we do. The plan is RARE will be for the 3D program, and C-RARE is what we're developing right now for our Type C observational study.
Thank you. Thank you. If we have any other questions, it doesn't look like we do right now. Thank you all so much for being here and sharing this information with the community. I know it certainly gives people a lot of hope to see all the work that's going on. So thank you.
Thank you.